Hello and welcome to this lesson for Advanced Higher Biology, which will be looking at sex determination. So what we're looking at is to understand how sex can be determined in different species. And you need to be able to explain how the presence of specific chromosomes can determine the sex of birds, mammals and some insects. Name the gene on the Y chromosome, which determines the development of male characteristics. Explain how the shape of the sex chromosomes can result in sex-linked patterns of inheritance. Explain what is meant by chromosome inactivation and how this prevents harm to individual females. Define what is meant by a hermaphrodite and how this can benefit individual members of the species. And give examples of environmental factors which can determine sex or sex ratio. So we are most familiar with species that have two separate sexes, males and females. The sex of an individual can be determined by either genetic factors or by environmental factors. When it comes to birds and mammals and some insects, the sex is determined by presence of sex chromosomes. These organisms are diploid, so have two full sets of homologous chromosomes. However, we've got one pair of chromosomes, which is a bit of an exception to the definition of a homologous pair. These are the sex chromosomes X and Y. Females have a homologous pair of X chromosomes, while males have one X and one smaller Y chromosome. During meiosis in the male, the X and the Y chromosomes pair up because there's a small area of homologous genes near the centromere. And meiosis ensures that there is one chromosome in each gamete. So all the gametes from an XX female will carry an X chromosome, and that means she is homogametic. All of her gametes are the same. For the XY male, half of the gametes are going to carry an X chromosome, and the other half carry a Y chromosome, and that makes him heterogametic. In most mammals, it's a gene called SRY, or sex determining region of Y chromosome, that is going to cause the embryo to develop male characteristics. So the default pathway in most embryos for development is female. And if you think about the fact that most males of most mammalian species have nipples, despite the fact that they will never produce milk or lactate in order to feed their young. This is because, as an embryo, they started to develop as female. The SRY gene present on the Y chromosome, though, is thought to act as like a master switch, and that triggers a cascade to activate all male developmental genes in the entire genome. So several of these genes will be on the autosomal chromosomes, so they will be present in females as well. However, they're only transcribed in males due to the control by this SRY gene. The X chromosome has got lots of genes on it which do not have homologous gene alleles on the smaller Y chromosome, and this leads to sex-linked patterns of inheritance. So the fact that if a female has two X chromosomes and the male has the X and the Y, the male is essentially missing some homologous genes on that Y chromosome. Sex linkage was first observed in Drosophila, the little fruit flies, and that's where they saw the inheritance of white eyes specifically. So in this case, we've got the dominant allele, big R, gives red eyes, whereas the recessive allele, little r, gives white eyes. So in females, a recessive allele R on one X chromosome can be masked by the dominant allele large R on the other X chromosome, and that makes them have the red-eyed appearance. So that's the heterozygous genotype shown as X big R, X little r, like this one down here, and is referred to as a carrier female. A male, on the other hand, has got a recessive R on the X chromosome, like this one here, is going to show up as having white eyes because the smaller Y chromosome has got no homologous gene to mask that effect. Red-eye males would only be seen if they inherit the X with a large R from their female. So because male cells have got only one X chromosome and female cells have got two X chromosomes, that might suggest that female cells are going to produce twice as much of the proteins from the genes on their two X chromosomes, but that's not actually the case. During early embryonic development, most of the genes on one X chromosome in each cell are inactivated, and that means the cells have only got a single working copy of the X chromosome genes, and this is going to vary from cell to cell. So if we consider this bunch of cells here, the inactivated one is the one that's greyed out, and the activated one is the one that's in colour. So at each cell, ones that are right next to each other, it might have been the X chromosome received from the mother that's activated, or it might be the X chromosome received from the father, and that's going to vary from cell to cell. So as mentioned previously, we've got some shared homologous genes on the X and Y chromosomes. So male individuals have got 2 copies of those genes. As a result, these are going to be left active on the inactivated X chromosome, and that makes sure that both males and female individuals have got the same number of active genes in their cells. So these chromosomes here that are inactivated will not be completely inactive. Some parts of it will in fact be active, as those are the parts that share the homologous genes that are present on the Y chromosome. And essentially that means that both male and female individuals are producing the same quantity of protein through the same amount of activity in their genes. So as the X chromosome inactivated in each cell is random, half of the cells in any tissue will have one copy of the gene expressed, and the other half will express the other copy. So if we consider a tortoiseshell cat, for example, it's got this genotype here. It's a heterozygote, so it's got the dominant allele and it's got the recessive allele. In this case, this one should be a small b. In this case, we have the dominant allele coding for black colour and the recessive allele coding for the ginger colour. So due to X chromosome inactivation, that means some of the cells will express the black fur allele and the rest will express the ginger fur allele. And that gives this kind of mixed appearance, essentially. So some of the cells, these ones here, are producing the ginger fur, and these ones here are producing black fur. So we have a mixed appearance in the individual, and that's a result of which chromosome has been activated in which cell. And that process lets carriers be mostly unaffected by any deleterious mutations on the X chromosome, as half of all the cells have got a working copy of the gene. Whereas in males, any deleterious mutations on the X chromosome that are inherited will show up. They're going to be more obviously seen as they don't have a working copy of the gene. Hermaphrodites, on the other hand, are species which have got both functioning male and female reproductive organs in each individual. So they produce both male and female gametes and usually have a partner with which to exchange their gametes. There's normally mechanisms available that means that individuals can't fertilise themselves. So, for example, many flowering plants would be considered hermaphroditic because they have both male stamens and female carpels available in the same flower, but normally they're not usually active at the same time. Land snails are also hermaphrodites because 2 individuals are able to entwine their bodies together and be fertilised by each other, so both individuals would be carrying fertile eggs. The benefit to the individual organism is that if the chance of encountering a partner is an uncommon event, so if they're not often going to see other members of their species, then there's no real requirement for that partner or that individual to be of the opposite sex in order for them to reproduce. There are also environmental factors which may influence sex determination or the ratio of males to females. So for example, Hermann's tortoise is a species where the incubation of eggs at less than 31 degrees produces all male offspring. However, above 32 degrees gives all female offspring. We may also see the activity of sex determination as a result of the members present in a specific group. So clownfish live in groups with one large female and some smaller males. However, if one of the large females is removed, the largest male then becomes a female. On the other hand, we also have the lesser mouse lemur. we have competition determining whether or not the ratio of offspring she has. So if our solitary female detects the urine of another female in her territory, she will automatically produce more male offspring in order to reduce her competition. And similarly, the Seychelles warbler will produce more female offspring when it's got a high quality territory. So when there's a lot of food available, more females will be present. Whereas when we're in a poor quality territory, more males will be produced. This is because males will usually leave the territory and will not remain to compete over resources. So there was a lot to get through there, but hopefully you can now understand how the presence of sex-specific chromosomes is able to determine the sex of birds, mammals and some insects. And that the gene on the Y chromosome determining the development of male characteristics is the SRY gene, which creates a cascade of transcription across the genome. That the shape of the sex chromosomes as a result of the Y chromosome being a lot smaller can result in sex-linked patterns of inheritance, as males do not have an extra copy of some of the genes present on the X chromosome. That chromosome inactivation occurs in individual female cells and occurs to let specific chromosomes, X chromosomes, be inactivated in each cell. So one X chromosome will be inactivated per cell, and that prevents harm to individual females as it prevents a double dose of the genetic material produced by the X chromosomes, and it also allows the masking of any deleterious alleles present on either X chromosome. That a hermaphrodite is an individual species which has both male and female reproductive organs and is able to produce both male and female gametes, and this can benefit individual members of a species as a result that they no longer need to necessarily look for a partner of the opposite sex. Any partner will do. And environmental factors which can determine sex and sex ratio would be temperature, for example in our Hermann's tortoise, and presence of competition and presence of resources.